Hey everyone, welcome to today's Explainer. I'm absolutely thrilled to have you all here. Have you ever actually stopped to wonder how life manages to remember its complex instructions over thousands, millions, or literally billions of years? It's a staggering achievement of nature. Okay, let's dive right in. Today, we are exploring the absolute evolutionary marvel that is DNA, the ultimate long-lasting blueprint for life. We're going to look really closely at the molecular architecture that allows DNA to survive and thrive, basically acting as an almost indestructible vault for genetic information. Get ready to understand biology on a truly foundational level.
So, here's our game plan for today. We'll start with the blueprint of life, then move into chemistry and missing oxygen, followed by thymine and error checking. After that, we'll look at building the twisted ladder, the ultimate backup system, and finally, wrap it up with a final review and pop quiz.
All right, part 1, the blueprint of life. Now, to really appreciate DNA, we first have to understand the fundamental problem that life faces, right? How do you store your most precious information securely? Imagine you have this highly confidential, massively important document. Would you write it on a flimsy paper napkin, or would you carve it into solid stone? Well, in the biological world, RNA is kind of like that paper napkin. It's a temporary, really fragile molecule, mostly meant to deliver short-term messages and then just get broken down. DNA, on the other hand, is the stone tablet. It's an incredibly robust, highly stable format designed for serious, long-term storage. But you're probably wondering, what exactly gives DNA this stone-like durability? That brings us to Section 2, Chemistry and Missing Oxygen. Let's learn a fantastic vocabulary word today, labile. When we say a molecule is labile, we just mean it's easily degradable, chemically reactive, or, really prone to breaking down. And RNA is highly labile. Why? Well, because every single nucleotide in RNA possesses what chemists call a reactive 2-OH group, which is an oxygen and hydrogen pair attached to its sugar ring. And this little oxygen atom makes RNA super reactive. It acts almost like a microscopic pair of scissors, giving RNA catalytic properties that allow it to just easily degrade and chop itself apart. So the key takeaway here is that this specific oxygen makes RNA inherently unstable. So how on earth did evolution solve this instability problem for long-term storage? Well, it performed a brilliant chemical upgrade. DNA, which literally stands for deoxyribonucleic acid, is deoxy, meaning it completely lacks that reactive 2'OH group. It just simply removes that troublesome oxygen. And by dropping that one specific atom, DNA becomes chemically way less reactive. It essentially loses that self-destruct mechanism, making it structurally far more stable over time. It's such a wonderfully simple but profoundly effective chemical tweak, don't you think? Moving right along to Section 3, thymine and error checking. Now, if you look at the genetic letters that make up the code, DNA and RNA share most of them, but they have one glaring difference. RNA uses a base called uracil, while DNA swaps that out and uses a base called thymine instead. So why make the switch? Well, swapping in thymine provides additional stability directly related to DNA's repair processes. You see, when a mistake happens in the genetic code, which definitely happens, the presence of thymine makes it so much easier for the cell's repair machinery to spot the error and fix it. Using thymine is honestly like writing with a pen that has built-in spell check. Okay, Section 4, building the twisted ladder. You can totally think of the DNA double helix structurally like a twisted ladder, or even a zipper. The two long strands are held together right in the middle by hydrogen bonds, and these act like the rungs of the ladder or the teeth of the zipper, keeping everything tightly clasped. But there's another absolutely crucial structural feature here called base stacking. Basically, the plane of one base pair stacks perfectly right over the plane of the other. Imagine laying solid bricks one on top of the other to build a sturdy wall. This stacking creates incredibly strong, stable internal forces that keep the DNA molecule rigidly in place and highly protected from the elements. And actually, mapping this exact physical architecture is what paved the way for all modern genetic research, eventually allowing scientists to sequence entire genomes and develop amazing tools like CRISPR. Which brings us to Section 5, the ultimate backup system. Because DNA is double-stranded with complementary base pairing, it effectively has its own built-in backup drive. If one strand gets damaged, the cell doesn't just lose the information, because the exact complementary code is sitting right there on the opposite strand. This dual-strand architecture allowed life to evolve highly sophisticated processes of repair. It's exactly like fixing a pothole on a broken road. The repair crew can just look at the undamaged lane to know exactly how to fill in and reconstruct the broken section. RNA, being mostly single-stranded, simply doesn't have this incredible luxury. And just look at how remarkably resilient this backup system actually is. Let's say you apply intense heat to a DNA molecule. Those hydrogen bonds will break, and the two strands will completely separate from one another, effectively unzipping the latter. Now, you might think the molecule is destroyed, right? No way! The moment you remove the heat and provide appropriate conditions again, those complementary strands naturally find each other and zip seamlessly right back together. It's this self-assembling, incredibly forgiving structure that just completely blows my mind. All right, Section 6, final review and pop quiz. Okay, class, get your mental pencils ready because it's time to test what we've just learned. Let's really lock in this knowledge by breaking down the summary table. On the DNA side, it lacks that reactive 2-OH group, uses thymine for error checking, forms a highly stacked double helix, and is supremely stable. On the RNA side, however, it retains that reactive 2-OH group, uses uracil, is highly labile and degradable, and even has catalytic properties that make it act like molecular scissors. Seeing it side by side like this really highlights exactly why DNA is the superior vault for our genetic code. Okay, question 1. Just shout it out in your head. Which molecule contains a reactive 2'OH group that makes it easily degradable and labile? Got it? It's RNA. That extra oxygen is the absolute culprit for its fragility. Moving on to question 2. What specific base does DNA use instead of uracil to help with error checking? Let's try question 3. What structural feature of the double helix acts like solid bricks in a sturdy wall? Remember? That's base stacking, where the plane of one base pair rests really securely over the other. All right, almost done. Question 4. What feature of DNA acts as a built-in backup drive, allowing it to repair itself, kind of like fixing a broken road? It is the double-stranded complementary base pairing. If one side breaks, the other side holds the exact blueprint to fix it. And our final question for today. If heat separates the strands of a DNA molecule, what happens when appropriate conditions are provided again? These zip right back together. They come right back together perfectly. And, well, that brings us to the end of our explainer. I really want to leave you with this one thought. Knowing how chemically resilient, highly stacked, and self-repairing DNA actually is, does it make you look at the unbroken chain of life on Earth over billions of years just a little differently? This robust microscopic vault is literally the reason we are all here. So keep asking questions, keep studying biology, and I will see you in the next explainer.